Welcome back to this next video in which we are discussing influenza. Okay, so we're currently in the process of discussing the adaptive immune response to the influenza viruses. So we discussed that in response to influenza, you're going to get two arms of the adaptive immune response being activated. You're going to get a B-cell response, also called a humoral adaptive immune response, which is a response to extracellular pathogens, and you're going to get a cytotoxic T lymphocyte response, which is a response to intracellular pathogens. Now, before we move on to discuss B-cell activation, just allow me to summarize what we have seen about T-cell activation. So both of these responses require T-cell activation initially. And firstly, I'll just summarize the intracellular one because we've completed that one now. We've completed this pathway. And then I'll summarize the extracellular pathway and we'll continue that on and we'll see how it then is going to result in B-cell activation. So the intracellular one first. So remember, everything begins with antigen-presenting cells. These are the cells that can activate T cells. And the two major examples are dendritic cells and macrophages. Now, if they want to activate the intracellular pathway, then they need to be aware that the pathogen is intracellular. How can they find the pathogen intracellularly? Well, they can do this by phagocytosing dying epithelial cells that are full of the viral antigens inside their cytoplasm. So when they phagocytose dying epithelial cells, they will chop up all the proteins in here, and that means they'll chop up all the influenza viral antigens, and because they found them inside one of the cells, they now know that this is an intracellular pathogen, and therefore they're going to try and activate the intracellular pathway, and to activate the intracellular pathway, they need to put the peptide fragments from the antigens of the pathogen on their surface bound to MHC class 1 molecules. So, overall, what we end up with is loads and loads of antigen-presenting cells that are all covered with MHC class 1 peptide fragment complexes, where you have a whole huge great variety of different peptide fragment complexes from all the different antigens of the influenza virus. So we are going to present to the T cells of the adaptive immune response loads of different peptide fragments from the influenza viral antigens. And again, I'll stress this really important concept that different antigen presenting cells will have cut the antigens up in different ways because it's random, so you will get a huge variety of different peptide fragments that you could have got from the influenza viral antigens. So an absolutely huge number of peptide fragments from influenza viral antigens are going to be presented now to T cells. Now the type of T cell that will respond to peptide fragments presented on MHC class 1 and which are involved in this intracellular response are the CD8 positive T cells. So remember, there are two populations of T cells, the CD8 positive T cells and the CD4 positive T cells. The CD8 positive T cells are involved in this intracellular response, whilst the CD4 positive T cells are involved in the extracellular response. They're involved in helping B cells to activate. So, each CD8 positive T cell is targeted against a different peptide fragment, and remember, in principle, your body should only contain CD8 positive T cells that are directed against peptide fragments that do not appear in any self-protein, and therefore can only be peptide fragments from antigens of pathogens. So what will happen is antigen-presenting cells will go to the lymph nodes and they will try and find CD8 positive T cells directed against one of the peptide fragments that they have on their surface. So what will now happen is you're going to get a huge number of CD8 positive T cells activated that are directed against some of the peptide fragments of the influenza viral antigens. A huge number of these peptide fragments will not be successful. You won't be able to find a CD8 positive T cell that actually is directed against that one. Maybe because it doesn't actually exist in the body, or because the CD8 positive T cell is far, far away in a lymph node that isn't draining lymph from the respiratory tract, so it's just in the wrong place at the wrong time. The point is, because we're presenting so many different peptide fragments of the influenza viral antigens, that some of them will at least successfully find a CD8 positive T cell that's directed against that peptide fragment, and therefore you're going to get them activated. So, what happens then is when a CD8 positive T cell is going to be activated by an antigen presenting cell with the MHC class 1 peptide fragment complex that it's directed against, what will happen is it will divide and divide and divide to produce a much bigger population of all identical CD8 positive T cells that are all directed against that peptide fragment, and then these will differentiate. Now, some of them, a small fraction of them, will differentiate into memory CD8 positive T cells, and these replace the original one that we used up to produce this massive population. You can't let them all become the effector ones, because the effector ones are short-lived, and then you'd wipe out this clone, so you could never then activate it again if you got infected by this pathogen again. Okay, so you have to replace the original one, and in fact you replace it with far more cells than you had originally, so you produce lots of these memory CD8 positive T cells that do the same thing as this original one and can be activated by antigen presenting cells presenting the peptide fragment to which they're directed against to them. And we'll do the exact same thing. They'll divide and divide and divide, and the cells can then differentiate again into effector or memory CD8 positive T cells. The bigger fraction of this population of CD8 positive T cells will differentiate into the effector CD8 positive T cells, also called the cytotoxic T lymphocytes. Now remember, these are directed against a specific peptide fragment, but you will have concurrent activation of these CD8 positive T cell pathways, so multiple different CD8 positive T cells directed against different peptide fragments of the influenza viral antigens will be being activated in this way. You'll have loads of these pathways being initiated, so you'll get loads of different effector CD8 positive T cells being produced directed against different peptide fragments of the influenza viral antigens. Now these go into the blood, they go to the site of inflammation, the respiratory tract in this case, and they are very, very good at killing virally infected cells. How do they recognize virally infected cells? Well, remember, all nucleated cells continuously sample proteins from their cytoplasm, chop them up into little fragments, and put those fragments on MHC class I molecules on their surface. And when they're virally infected, that means that they will end up putting peptide fragments from the influenza viral antigens on their surface bound to MHC class I, and if this cytotoxic T lymphocyte finds the MHC class I peptide fragment complex that it's directed against, it will bind to the cell, and then it will kill that cell through perforin granzyme mechanism and fast ligand mechanisms. Okay, so this is the cytotoxic T lymphocyte response, and overall it kills virally infected cells. It will go to the respiratory tract and get rid of a huge number of virally infected cells, cause them to commit apoptosis, and when they commit apoptosis, all of the influenza virus stuff inside them will be destroyed. So the cell dies to save other cells, so it sacrifices itself on the orders of the cytotoxic T lymphocyte. So that's the cytotoxic T lymphocyte response complete. That is it. That is that arm of the adaptive immune response, the intracellular arm, killing intracellular pathogens, the pathogen whilst it's intracellularly. Let's now go on to the extracellular response, which we did not finish. We just saw the T cell activation. So we need to go back up here. Right, so antigen-presenting cells start the thing off again. This time they're going to phagocytose the pathogen whilst it's extracellularly to initiate the extracellular pathway. So they'll phagocytose influenza viruses just floating around in the extracellular fluid. They'll break down their antigens into peptide fragments, and because they found this extracellularly, they will say, let's initiate an extracellular response. So we need to put the peptide fragments that we get from its antigens on MHC class II molecules this time. So this time, the antigen-presenting cell will end up covered in MHC class II peptide fragment complexes, and again, it will end up with a huge number of different peptide fragments from the different influenza viral antigens, and you'll have more than one antigen-presenting cell doing this, and they'll cut up the antigens in different ways, so again, we'll be presenting a huge scope of different peptide fragments to the adaptive immune response. Coming down, the antigen-presenting cells will go to the lymph nodes, and here they're going to try and activate CD4 positive T cells, which are the cells of the extracellular response. Again, all of the different CD4 positive T cells are directed against a different peptide fragment, and you should only have them directed against peptide fragments that are never in any self-proteins. In the ideal world, anyway, that's the way it will work. That's the idea of it. So what will happen is when the antigen-presenting cells actually find a CD4 positive T cell directed against one of the peptide fragments that they're presenting on MHC class 2, they will activate that CD4 positive T cell, and again it does the exact same thing as we've been through for the CD8 positive T cells. It divides and divides and divides to clone and expand. Some of the population will then become memory CD4 positive T cells and will replace the original one, so that if we ever want to initiate this again, we have, you know, some of these cells that can do that. And then others will differentiate into effector CD4 positive T cells, also known as helper T cells. Now remember, there are different types of effector CD4 positive T cells or helper T cells. 4 major different types: T helper 1, T helper 2, T helper 17, and T follicular helper. And remember, it's not particularly well understood what it is that determines which type of helper T cell these CD4 positive T cells here are going to differentiate into. What we think happens, however, is that all the cells at the site of inflammation release molecules, inflammatory mediators, signaling molecules that will go into the lymphatic vessels, drain to the lymph nodes, and instruct these CD4 positive T cells what type of effector T cell is necessary to conquer this type of pathogen. Now, in the case of influenza virus, the major type that we're going to form is the T follicular helper, because this is the one that's most diligent at activating the B cell response, which is what we want. All of them can activate the B cell response, but these other types do more than just activate a B cell response. They do other things as well, and they're used in fancier different types of infections. So remember, when you have macrophages struggling, you want T helper 1 cells. When you have a multicellular parasite, you want T helper 2 cells. When you have fungal cells, you want T helper 17 cells. None of these are relevant to us. They are 3 other arms of the adaptive immune response that I've discussed to give you a fuller picture of the adaptive immune response, but which are not relevant to us here. So the type that is relevant to us is this type here, the T follicular helper cells, which are fantastic at helping B cells activate, which is what we want in influenza virus. We do not need macrophages to receive help from T helper 1 cells. They are not struggling to destroy influenza virus particles the way they would be in Mycobacterium tuberculosis. We certainly do not need eosinophils to attack the influenza virus. That would be totally over the top. And we don't need loads and loads of neutrophils either, because we're not dealing with a fungal cell. So we do not need these other 3 types of effector CD4 positive T cells. So we just want this one so that we can get a nice B cell response initiated. All right. Okay, so we now need to discuss B cell activation. So I think I will go over here. We'll put this as the title. So we're now going to discuss B cell activation, and we'll see how the T follicular helper cells are going to be really important in helping, in assisting with the activation of B cells. And indeed, this is why helper T cells are called helper T cells, because they help B cells to activate. Okay, so B cells are the other major type of lymphocytes. So of the lymphocytes, the 3 major types are the B cells, the CD4 positive T cells, and the CD8 positive T cells. Now, that means that B cells will also hang around in the blood, the lymph nodes, and also the spleen. And you have a huge number of different B cells. And the B cells, like the T cells, they are specifically targeted against specific molecules. So I'm just going to draw a picture of a B cell so that I have something to point us here, and I think we'll do it in green. So again, it's going to be a classic lymphocyte cell, which is nice and spherical and has a very large nucleus here. Now, B cells then, they all have a specific design of antibodies. So remember, I'll just draw another picture of an antibody because I don't want to have to try and find the picture I drew ages ago of an antibody, so I'll just draw it out again. Remember, antibodies are made out of 4 protein molecules bound together, 2 identical heavy chains, and then 2 identical light chains bound to the heavy chain. So here is one heavy chain, here's another heavy chain, they're bound together there, and then each of them has a light chain bound to them. And these produce binding sites for antigens at the tip here. Now, each of these B cells produces its own design of immunoglobulin molecule like this. So it produces its own immunoglobulin heavy chains, its own immunoglobulin light chain design, and therefore it has its own antigen binding site here. So all of the different B cells are targeted against different antigens. And more than that, they're targeted against different epitopes of an antigen. So if I just draw some antigen here, this blob here is going to represent an antigen. So this is an antigen. The antigen has loads of little facets you can see here, and those facets are known as the epitopes of the antigen. So the little domains of the antigen to which an antigen binding site could bind. And you know, this binding site, if it binds to this epitope, it's not going to be able to bind to this epitope or this epitope. So the antibody molecules aren't just specific for an antigen, they're specific for an epitope of an antigen. So, in our body then, we have a huge number of different B cells, all with their own design of immunoglobulin, and therefore they are targeted against different epitopes of antigens. Now again, in principle, you should only have B cells with an immunoglobulin design that binds to epitopes that are never found in any self-proteins, so that we only have B cells that are directed against pathogen epitopes. Okay. So let me now go over how B cells are going to be activated as part of the adaptive immune response. So we have to go back to the site of inflammation. Remember, at the site of inflammation, cells are going to be dying, and I'll just actually draw this here. We'll draw a little flow diagram again. So let's say here we have a ciliated columnar epithelial cell that is infected with influenza. So I'll draw a red cross for it. So this is an infected cell. Now, we've been over the fact that these virally infected cells will eventually die because of the virus infection. And when they die, they're going to release a lot of molecules into the extracellular fluid. And of course, this was really important in initiating the inflammatory response because they release loads of DAMPs into the extracellular fluid, the damage-associated molecular patterns, which is what the sentinel cells use to know that there is a serious problem and what makes them start the inflammatory response. However, they're also going to release a whole bunch of the influenza virus proteins into the extracellular fluid, because remember, this cell was full of influenza viral antigens. It had in its membrane loads of influenza viral antigens, the hemagglutinin proteins, the neuraminidase proteins, the M2 ion channel, the MB ion channel if we're talking about an influenza B virus, or the hemagglutinin esterase fusion if we're talking about influenza C virus. The point is they were full of influenza viral antigens, and those are now going to leak into the extracellular fluid. So, when the cell dies, this is going to release the influenza antigens into the extracellular fluid. So, what's now going to happen is some of these influenza antigens will now drain in the lymphatic fluid to the lymph nodes. And I think I'll get another colour. I'm getting bored of this evil red colour. So, these influenza viral antigens are now going to go to the lymph nodes that drain the respiratory tract, the same lymph nodes that the antigen-presenting cells that were going to activate the T cells have gone to. And now it's the actual antigens coming themselves to the lymph nodes that are going to activate the B cells. So you're going to have a huge number of these influenza antigens coming, and all the different antigens are going to be coming. So all the different proteins of the influenza virus, they're going to be coming. They have a huge number of different epitopes. They're draining to these lymph nodes. These lymph nodes are full of B cells, and the B cells have on their surface their immunoglobulin, their specific immunoglobulin, a special form of it with membrane anchors so that it's bound to the surface of the cell. And let me just draw this on over here. So I'll do this in normal blue. So on the surface of the B cells, they produce a special form of their immunoglobulin. So this is the immunoglobulin molecule. Now, they can make a special form of it with extended bits here that means that it can anchor itself in the cell membrane. And when they do this, it's known as the B cell receptor. So this is the BCR for short. Now, this is actually strictly the difference between what we mean when we say an immunoglobulin molecule and an antibody molecule. Immunoglobulin is actually a slightly broader term. We would still describe the B cell receptor as the immunoglobulin molecule, just with these special membrane anchors, whereas an antibody specifically means a soluble immunoglobulin molecule, a form that doesn't have these membrane anchors and therefore is just floating around in the extracellular fluid. So I hope you understand that. This term immunoglobulin covers both the B cell receptors and the antibodies, and although they are identical to one another as far as this antigen binding site up here is concerned, they're not identical to each other as far as the membrane binding bits are concerned that the B cell receptor has, but the antibody molecules must not have, otherwise they'd have been membrane bound themselves. Okay, so B cells have on their surface loads of copies of their special design of immunoglobulin molecule with this special modification that means that it's going to be implanted in their cell membrane, and these are called the B cell receptors. So now, hopefully you might get what's going to happen here. We're bringing loads of the influenza viral antigens to lymph nodes. Those have a huge number of epitopes. In the lymph nodes, there are going to be B cells with B cell receptors on their surface, and some of those B cells in the draining lymph nodes that drain the respiratory tract will happen to be the right B cells. They'll have the right design of immunoglobulin molecule that one of the influenza viral antigens epitopes can bind there. So, let me draw this on our flow diagram. So what's now going to happen is here we have our B cell, like so, again, the spherical cell with the large nucleus, and this is going to be a B cell with a design of immunoglobulin that can bind to one of the epitopes of an influenza viral antigen. And again, remember there are loads of these antigen molecules coming to these lymph nodes. Each antigen molecule has a huge number of epitopes. Most of those epitopes will not actually successfully find a B cell with a immunoglobulin design that's directed against it, but some of them will, and therefore concurrently we're going to be activating loads of B cells in these lymph nodes that are directed against different epitopes of the influenza antigens. So we're going to follow it for one of these B cells, but remember that this pathway will have lots of concurrent pathways going along with it that are B cells directed against different epitopes of potentially different influenza antigens. So I hope you understand that. Again, I'm showing one of these pathways going on, but remember there's multiple of them going on concurrently. Okay, so what's going to happen then? If this B cell has a design of immunoglobulin that is directed against an epitope of the influenza viral antigen, and that influenza viral antigen comes to it, what's going to happen is the influenza viral antigen is going to bind to the B cell receptor on the surface of the cell. So let's just put this on here. So here is the B cell receptor, drawn here, and let's have the influenza viral antigen again here in red. There's its epitope that is binding to the B cell receptor, and obviously I don't know what this is representing. None of the influenza viral proteins look like this, but it's just a drawing. This is representing one of the influenza viral antigens, and I've shown it in this ridiculous shape just to sort of highlight the concept of an epitope being this special domain that fits into that binding site of the immunoglobulin molecule. Okay, so when this happens, this B cell is obviously one of the B cells that we want to activate. It has an immunoglobulin design that is against one of the influenza viral antigens epitopes. So we want to activate this. Now, you might think that what should happen now is this B cell should divide and divide and divide. We should get a huge population of the B cells. Some of the B cells should then differentiate into effector B cells. Some, of course, should differentiate into memory B cells to replace the original one, because the effector B cells, again, are not long-lived. We need to replace the original one so that we can initiate the immune response again. And that's what should happen. However, that is not what happens. B cells are not given that right to do that. In order to activate, they have to go through a checkpoint. They have to receive help from helper T cells in order to activate. And this is really a powerful checkpoint. It's to make sure that we are not initiating B cell responses that are against self-proteins. Now, I told you that in principle we should not produce B cells that are directed against epitopes of self-proteins. However, sometimes that goes wrong. In order to try and prevent these B cells that are directed against self-proteins ever actually being activated, in order for B cells to be activated, they have to not only find their actual antigen and bind to it, but they also have to receive help from a helper T cell that is directed against a peptide fragment of this antigen that they are directed against. And this is a security mechanism. This is to make sure that you do not activate B cells unless you have activated T cells. So it's a checkpoint because it means that if you were to activate B cells against a self-protein, it would have to mean that both the B cells had gone wrong and you ended up producing a B cell directed against a self-protein, and the T cells had gone wrong, you'd ended up producing a T cell against a peptide fragment of a self-protein as well. And it's less likely that that will happen, that T cells and B cells will go wrong, than just the B cells going wrong. Of course, sometimes in the autoimmune diseases, it does end up going wrong, but autoimmune diseases are rare, so usually it doesn't happen. So this is a checkpoint. It means that the T cell response and the B cell response have to both be there in order to actually get the B cell response. So, how is this B cell then going to get help from the follicular helper T cells that we've discussed earlier on? Well, what do you think it's going to do? Remember, the effector T cells, the T follicular helper cells, they have on their surface a T cell receptor that is directed against MHC class II with a peptide fragment. Let's say that we have launched an T cell response against peptide fragments of this antigen, which we probably will have. This is one of the influenza viral antigens. We have chopped it up into loads of different peptide fragments, loads of different times, so that we have a huge number of the different possible peptide fragments that you could get from this being presented to CD4 positive T cells. So the likelihood is that we will have produced some CD4 positive T cells, some T follicular helper cells that will be directed against peptide fragments of this antigen. So they are going to help the B cell if it can demonstrate that it is directed against an antigen which they are directed against the peptide fragment of. So what the B cell needs to do is it needs to internalize this antigen, chop it up into peptide fragments, and put those peptide fragments on its surface bound to MHC class II molecules, and then it can receive help from the follicular helper T cells. So that's what it's going to do. It will endocytose this, so this will come into a little endocytic vesicle. It'll break down this antigen into loads of tiny little peptide fragments, and it will put those peptide fragments on its surface bound to MHC class II, which I'll just put in here. So here is, of course, the MHC class II molecule, and bound in the peptide binding groove there, we're going to have a peptide fragment. And of course, this is showing one of those. In reality, you'll have loads of them. Now, also remember that this B cell is covered in B cell receptors. Loads of the influenza viral antigens are coming to the lymph node from the respiratory tract. So actually it's going to get loads of its B cell receptors binding to the antigen. In fact, this B cell receptor could bind to two of the antigens at once because it does have two antigen binding sites. So the point is this B cell is going to endocytose loads of these antigen that it's directed against an epitope of, and therefore it'll cut loads of them up into peptide fragments. It does the cutting up randomly, so that my point here is that it will cut the different antigens up in different ways, and therefore will end up with a huge number of the different possible peptide fragments of this antigen, which it will then have on its surface bound to MHC class II molecules. And the chances are now that this will end up putting on its surface a peptide fragment of this influenza viral antigen to which we have produced a follicular helper T cell that is directed against this. And the follicular helper T cells will also be in the lymph nodes, which is where this B cell is. So let me now put this in. So what colour should we have the follicular helper T cell? We'll have it in yellow here. So here, this is representing our follicular helper T cell. And I'll just try and put a little arrow in here. So here over here. This is our follicular helper T cell. It's a spherical cell with its great big nucleus here. And of course, on the surface of that cell, you'll have the T cell receptor, which will be directed against this MHC class II peptide fragment complex that we've got here, or will be directed against one, hopefully, of the MHC class II peptide fragment complexes that this B cell has put on its surface. So this here is the T cell receptor, and that cell is a T follicular helper cell. So, if the follicular helper T cell comes over to this B cell and finds that the B cell has on its surface the peptide fragment that it is directed against, it says, "Weepy, you are evidently a good B cell. You are directed against an epitope of the antigen that has the peptide fragment that I am directed against. That means that we are on the same page. You are attacking the same antigen that I am attacking, and therefore you can you can activate. You are safe to activate." So it then says, B cell, activate. And this is how helper T cells help B cells. Now, as I say, it is not just the follicular helper T cells that can do this. The T helper 1 cells, the T helper 2 cells, and the T helper 17 cells, they all have the T cell receptors on their surface. They can all do this. The other ones just do other things on the side, fancier things as well as doing this. Therefore, not as many of them will stay in the lymph node where the B cells actually are. Follicular helper T cells don't go into the blood, they don't go to the site of inflammation, they stay in the lymph nodes to help the B cells. So they're the most diligent, whereas these ones, they can bugger off effectively and go into the blood and then go to the site of inflammation. Okay, so they're not as diligent at doing it because they have these other jobs to do as well. Right, so what happens then when the follicular helper T cell says to the B cell, you can activate? Well, of course, now it's the bit that we were waiting for. The B cell is going to divide and divide and divide. We want to expand its clone, want loads of B cells all directed against this epitope of the influenza viral antigen. So I'll draw these all in here. Here they all are. And of course, I'll just draw, let's say, 6 of these. In reality, you'll get absolutely loads of them. So the next thing that happens is division, and then what's going to happen is they're going to start to differentiate. Now, when they differentiate, they can differentiate into 2 different types of cells, the effector B cells, and then of course the memory B cells. So let's just put on memory B cells, because hopefully we have a very good understanding of memory lymphocytes now. So some of them will become memory B cells. So I'll just have, let's say, 2 of them becoming memory B cells. So a small fraction of the population that you overall produce will now become memory B cells, and those will replace the original B cell that we started off with, which was activated in this B cell response. Again, I'll give you the motivation for this because it's so important to understand. If all of these now became effector B cells are not that long-lived, so they will die pretty quickly from the body, and then we will have lost this B cell clone. We will have lost B cells with this design of immunoglobulin that is directed against this epitope that actually is an epitope of an antigen of a pathogen that has infected the body. We do not want that, because then we could never initiate that same B cell response again. So we must replace the original B cell, and we replaced it with more cells than you had originally. And this is what these memory B cells are. They replace this one, and if you want to initiate the response again, if you ever get infected by the influenza virus again, the same thing will happen. Influenza antigens will drain to the lymph nodes, and now sitting in those lymph nodes waiting will be lots of these memory B cells, all with this design of immunoglobulin on their surface. They will be activated in the same way, and they will do the same thing. They will divide and divide and divide, produce a massive great population, some of which will become effector B cells, and some of which will then produce memory B cells to replace them. So it goes round and round and round. So I hope you do completely understand what memory lymphocytes are all about now. So let's now talk about the other type of cell that they can differentiate into, which is, of course, the effector B cells, and more of them are going to become effector B cells than, of course, will become the memory B cells. So here we are. Here are some of the effector B cells. Now, effector B cells have another name. They are also called plasma cells, and in fact, plasma cells is the name that we use more often for effector B cells. So, effector B cells also called plasma cells. Okay, now what are these cells going to do? Well, they're going to start producing a huge number of the immunoglobulin molecules that are soluble. So they're going to produce a huge amount of the antibody molecules. And I want to apologize, I have actually drawn these slightly wrong. They're no longer spherical in the way that the other lymphocytes are. Plasma cells are actually more of an egg-shaped cell like this. So this is a better picture of a plasma cell. So imagine I could rub these out. I won't actually bother rubbing them out, but imagine I can rub these out and replace them all with these egg-shaped cells here. And the reason that they have this shape is that this space in the cytoplasm here is the extra space that they now need to start producing all of these antibody molecules that they're going to start producing. And they really are little antibody factories. They produce thousands and thousands and thousands of these molecules. And that's what this extra bit of cytoplasm that makes them an egg-shaped cell is necessary for. Okay, so a lot of these effector B cells will become plasma cells, and what do the plasma cells do? They're going to leave the lymph nodes, they're going to go into the blood, and then they're going to move out of the blood into the bone marrow. They actually like sitting in the bone marrow. So I'll just draw a little picture of a long bone. We'll have a picture of the femur here. So they go into the bone marrow. So there is the head of the femur. Here's the shaft of the femur. And here are the condyles of the femur at the other end. So they go into the bone marrow, and they sit in the bone marrow, which of course is the tissue at the centre of bones, at the centre of the long bones. So it's this portion of tissue at the centre of the long bone, this area here. They sit in the bone marrow, and from the bone marrow they will start producing loads of antibody molecules. These antibody molecules will be secreted into the extracellular fluid, they'll go into the bloodstream, and then they can circulate in the bloodstream to the site of infection. They can leave the bloodstream there and help destroy the pathogen. Okay, so that's what plasma cells are going to do. There is another complication with regards to plasma cells, which is that different plasma cells can secrete different types of antibody molecules. So there are different types of soluble immunoglobulin molecule that we need to go over. Alright, so let's just talk about this. So these produce antibody molecules, which for short I will abbreviate down to ABs, like so, antibody molecules. Now, antibody molecules, there are different types of antibody molecules that we use against different types of pathogen. And these, the four main ones are IgG, IgA, and IgM and IgE. So IgG, IgM, IgA, and IgE. So the next thing that I want to explain is what is the difference between all these different types of antibody? What is the difference between what they're going to do? Okay, so when these B cells here are deciding to differentiate into a plasma cell, which looks like this, they will also pick a type of antibody molecule that they are going to actually produce. They will pick either IgG, IgM, IgA, or IgE. And in response to influenza, the two main types that we're going to get plasma cells producing are IgG and IgA. We will get a little bit of IgM produced. We will not get IgE produced. So, what's the difference between these molecules? Well, they do have slightly different lower portions of the molecule. So, coming back to this picture of an immunoglobulin molecule here, up at the top is where the antigen binding site is, which is where they bind to a certain epitope of the antigen molecule. The lower portion of the immunoglobulin molecule, however, that's not involved in binding to the epitope of the antigen molecule. So that is not a portion that the B cell will have a unique design of. So I talked about how each B cell that you've got in your body has a unique design for the immunoglobulin molecule. The bit that varies is these bits right at the tips of the heavy chain and the light chain, not these bits, the lower bits. These bits are constant between different B cells. However, once a B cell is differentiating into a plasma cell, what it can do is it can say, okay, I've got my special design of antigen binding site, but there are different options for this constant region of the immunoglobulin molecule down here. And I'll just sort of chop this bit out. So I'll just highlight this bit by boxing it around like this. So this portion down here is not the bit that's involved in binding to the antigen molecule. This bit is known as the constant region, or the FC region, the fraction crystallizable, the constant region. So this is the constant region of the immunoglobulin molecule. It's not the portion that is directed against a specific epitope of an antigen that is a unique design for the B cell, whereas these bits up here are the antigen binding site. They're the bit that the B cell actually has designed itself. Now, when the B cell has actually differentiated into a plasma cell, it takes the decision of what type of immunoglobulin molecule it wants to secrete into the body fluids, and it can change this constant region. There are different types of constant region it can put here and have its specific design of antigen binding site attached to. And this is how these different types of antibody molecule are different. IgG will have a different Fc region to IgM, which will have a different region to IgA and IgE. All of these will have different Fc regions. So, if we were to produce 4 plasma cells then, let's say, and each of those 4 plasma cells chose to produce a different type of the antibody molecule, then what would be different between the antibody molecules is the Fc region. They'd all produce antibody molecules with the identical antigen binding site because that's the set design of antigen binding site that we wanted. So they all have the same antigen binding site, but they would have different Fc regions, and that means that what the antibody molecules can actually do once they've bound to their antigen is different. The effector function of the antibody molecule is different. However, the antigen binding site, the antigen specificity is exactly the same. So I hope I have nailed down that point. So, when we are producing these effector B cells, these plasma cells, they take the decision of what type of antibody molecule they want to be producing. They take that decision here. They can't then change that decision. They make that decision when they differentiate. Now, what tells them what decision to make? Again, we do not understand that. It's like what determines what type of CD4 positive T cells you're going to get, the effector types of CD4 positive T cells. We have speculation. We think that in that case it is molecules being released by the inflammatory cells at the site of inflammation which drain to the lymph nodes and inform the CD4 positive T cells that are about to differentiate what type of pathogen we're dealing with and therefore what type of helper T cells we're going to need. Here we think the idea is exactly the same because these different types of antibody molecule are useful against different types of pathogen. So we think this is going to be determined by signaling molecules that are released by immune cells at the site of inflammation, which will drain to the lymph nodes, which is where the B cells are differentiating now into plasma cells, and will determine what type of antibody molecule the plasma cells, once they've differentiated into plasma cells, will produce. Okay, so that's how they decide, or our speculations of how they decide. Let me now talk about what these different antibody molecules are good at doing. So IgG, this is the main type of antibody molecule produced always in B cell responses. This is a nice simple antibody molecule. It functions as a monomer, which is not the true of all these different types of antibody molecule. So here is a nice little picture of IgG, and because it functions as a monomer, like so, it can easily get out of blood vessels at sites of inflammation and therefore will go into exudate. In addition, it also has a way of coming out of blood vessels, capillaries, and postcapillary venules, even at sites where inflammation is not present, and therefore it can circulate in the extracellular fluid all over the body. So IgG is very good at going all over the body into the extracellular fluid, the ECF for short, and looking for the antigen molecule that it is directed against. So IgG goes all over the body in the extracellular fluid. In contrast, IgM does not. IgM is a much bigger molecule because it binds into a pentamer, and I'll just try and draw a little picture of this, although it's doomed to go wrong. So there's one, then let me put another one in, and here it's already starting to get too cramped. So here's another one down there, then let's have another one down here, like so. So that's the third immunoglobulin molecule. Let's put another one in up here, like so. And one more like that. Okay, so IgM, it binds into this pentameric arrangement, which IgG does not. It does not form pentamers like this. So IgM forms pentamers like this, and this means that IgM forms much bigger molecules than IgG. And then when you're a massive great protein molecule like this, you're going to struggle to leave blood vessels. So IgM generally stays in the blood. So it mediates blood-borne immunity. So it's going to go around in the blood, and if it finds any antigen molecules that it's directed against, that its antigen binding site is directed against, it will bind to them and destroy them, attack them. So it is one that stays in the blood. So IgM is the one that you produce if you need the antibody molecules to stay in the blood, whereas IgG leaves the bloodstream and goes into all the extracellular fluid. I mean, a bit of it will stay in the blood, but a lot of it will go into the extracellular fluid, both extracellular fluid that's exudate and extracellular fluid at sites where there isn't an inflammatory response. So it goes all over the body to look for the antigen molecule that it's directed against. IgA next. IgA forms a dimer. So here's one, and then here is the other one bound to it in a dimeric arrangement like so. So this forms a dimer. Now, what does IgA do? What's it used for? Well, IgA is the special one which is secreted onto loads of different epithelial surfaces. So this is a special type of immunoglobulin molecule. We secrete this from epithelial surfaces. So of course it will go into the blood initially, but then epithelial cells are very good at taking it out of the blood and secreting it onto epithelial surfaces. So it will end up on the epithelial surface that lines the respiratory tract. It will end up on the epithelial surface that lines the gastrointestinal tract, the epithelial surface that lines the urogenital tracts as well. So it ends up lining all the different epithelial linings, okay? And that's very, very useful, especially when the pathogen enters through an epithelial lining, because it means that we can have these molecules on that epithelial surface that can attack the pathogen. So obviously influenza virus infects an epithelium, so IgA is going to be an extremely important type of antibody molecule for us to produce, because it's going to be secreted onto the respiratory epithelium that lines the respiratory tract, and therefore is going to be able to attack influenza viruses at the respiratory epithelium. Now, the final one here of the soluble forms of antibody molecules, and I'll just find some other colour to do it in, is IgE. This one's not relevant to us at all. This is one that's used in response to multicellular parasites again. So this is a parasite response. So just like T helper 2 cells were a parasite response, IgE is a parasite response. And the reason this is a parasite response is that what this can do is it can bind onto the surface of the parasites, obviously, because it can bind to the antigen that it's directed against, which may well be on the surface of the parasite, and then it directs the eosinophils towards the multicellular parasite. So remember, the story as we got up to was the T helper 2 cells come into the area of inflammation. Then when they find macrophages that have on their surface peptide fragments of antigens of the multicellular parasite bound to MHC class 2 that the T helper 2 cells are directed against, the T helper 2 cells realize they've come to the right space, and then they say, okay, we'll call for eosinophils. So they release interleukin-5 and interleukin-30, and the eosinophils then come into the site of inflammation. But then I ended the story at that point. I said, well, actually, no, I didn't end the story at that point. I said they'll go over to the multicellular parasite and they'll release their horrors on the multicellular parasite. But how do they then find the multicellular parasite? Here is the answer. IgE is important for directing eosinophils towards the multicellular parasite. So, just drawing a little picture of this. If this is our worm here, our multicellular parasite, what can happen is an IgE molecule can bind onto the surface of the parasite, and then eosinophils have receptors on their surface for the Fc region of IgE molecules. So I'll just draw this in here. So here comes the eosinophil, like so. Here is its bilobed nucleus, like so. I'll just colour that in a little bit. Okay, so this is our eosinophil. I'll just label it up. So on the eosinophil surface, there is a receptor for the IgE molecule here, and it binds to specifically that constant region of the IgE molecule, whereas it doesn't bind to the constant region of IgA, IgM, or IgG. So IgE is the one that we specifically use to direct eosinophils towards multicellular parasites, and then they will release their horrors, the eosinophil cationic peptide and major basic protein, onto this multicellular parasite. So IgE then is one that we produce against parasites. Okay, right, so the two major ones that we're going to produce in influenza viral infection then are IgG and IgA. We'll produce a little bit of IgM, we do not produce IgE. So we're going to get plasma cells that produce IgG, we're going to get plasma cells that produce IgA, and we'll get a few plasma cells that produce IgM. What then is going to happen is IgG molecules will go into the exudate or at the sites of inflammation. So they'll end up in the exudate at the respiratory epithelium. And the IgA molecules, they will actually be secreted by the epithelial cells onto the epithelial surface. The IgM that you produce will then remain in the blood and will neutralize any of the antigens that have ended up in the blood. So, how is this actually going to help them with dealing with the influenza viral infection? But before we actually ask that question, let me just summarize what we've seen of the B-cell response, and then we'll answer that question of how do antibody molecules actually help us to clear the viral infection. So we have now seen the B-cell response, and remember the way that this is activated is when the respiratory epithelial cells are dying because they've been infected by influenza virus particles. They will be releasing a huge number of the influenza antigens into the extracellular fluid. These will drain through the lymphatic vessels to the lymph nodes, and these are going to be the things that are going to activate B cells. So remember, in our body we have a huge number of different B cells that are directed against different epitopes of proteins, and we should only have B cells that are directed against epitopes of non-human proteins, i.e. we should not have any B cells that are directed against epitopes of self-proteins. Now, what will happen then is when the influenza viral antigens arrive in the lymph nodes, they will come into contact with some B cells potentially that are directed against epitopes of them. So you will, hopefully, in those lymph nodes that drain the respiratory tract, have some B cells that are directed against epitopes of the influenza viral antigens that are arriving there. So, here is an example of a B cell that has an immunoglobulin design that is directed against an epitope of one of the influenza antigens, and we follow the pathway for this B cell. However, concurrently, there'll be lots of other B cells directed against different epitopes of potentially different influenza antigens that will also be undergoing activation. Now, in order for a B cell to activate, it needs to receive help from a helper T cell, and this is a checkpoint because for this B cell to activate, you have to also have had T cell activation of T cells that are directed against peptide fragments of the antigen that the B cell is directed against an epitope of. So the B cell then endocytoses the antigen that it has bound to its B cell receptor, chops it up into loads of little peptide fragments, puts those peptide fragments on its surface bound to MHC class II so that it can present the peptide fragments of the antigen that it is directed against to helper T cells. And remember, in influenza virus, we're going to get mainly follicular helper T cells. So, these helper T cells will then come along, and let's say this is a helper T cell that is directed against one of the peptide fragments of this antigen, and it will find the MHC class II peptide fragment complex that it's directed against on the surface of this B cell. And then it will say, right, we're on the same page, you are directed against the same antigen that I am directed against a peptide fragment of, so clearly we're doing the right thing here, you can activate. So it gives the B cell permission to activate. What will then happen is the B cell will divide and divide and divide, so you'll get a big population of B cells. Some of them will differentiate into memory B cells, which will replace the original one that you had so that you can launch more adaptive immune responses against that epitope of the antigen in the future. And the others will differentiate into effector B cells, which are also called plasma cells, which are an oval-shaped cell here. When they differentiate into plasma cells, they have to take the decision of which type of antibody molecule they're going to secrete, and there are these four major types of antibody molecule that they can choose to secrete. IgG molecules, which are monomers and which go into the extracellular fluid all over the body. This is the major type of antibody molecule. IgM, which is a pentamer, well, which forms a pentamer like this, and which mainly stays in the blood of the body and therefore mediates immunity in the blood. IgA, which is a dimer and which is secreted onto epithelial surfaces and mediates immunity of epithelial surfaces, so clearly that one's going to be really important in influenza. And then IgE, which is one that we use against multicellular parasites. So, how do these B cells decide when they're differentiating into plasma cells which type of antibody molecule they're going to commit to making? Well, we believe the decision is influenced by signaling molecules that are released by the cells of the immune system at the site of inflammation, just like we believe that the differentiation of CD4 positive T cells into a certain type of helper T cell is determined by signaling molecules that are released by immune cells at the site of inflammation and which are going to go to the lymph nodes through the lymphatic systems. So, in response to influenza, we get a lot of plasma cells that can produce IgG and a lot of plasma cells that can produce IgA, and of course we're going to get plasma cells that are producing these types of antibodies, and we'll have plasma cells that are directed against different antigens, different epitopes of the antigens of influenza virus. So remember, we are not just producing one type of plasma cell that can produce different types of antibody. When we're not just producing one type of plasma cell directed against one epitope of an influenza antigen. We're going to be producing loads of different clones of plasma cells that are producing antibodies directed against different epitopes of influenza virus antigens, and in each of those clones, we're going to have some plasma cells that can produce IgG, some that can produce IgA, and some that can produce IgM. Okay, so these plasma cells will go to the bone marrow where they like to hang out, and they will throw the antibody molecules that they're producing into the extracellular fluid where they'll go into the bloodstream, and then if they're IgM, they'll stay in the bloodstream. If they're IgG, they'll go all over the extracellular fluid in the body, but particularly they're going to end up in the exudate at the site of inflammation, and if they're IgA, they'll be secreted onto the epithelial surface. Okay, so that summarises B-cell activation, and therefore we have almost finished the adaptive immune response. What I now need to tell you is why are antibody molecules so important? How are these going to actually help us? So overall, we have produced loads of IgG, loads of IgA, and loads of IgM, and if we were to look at all the different IgG molecules and IgM molecules and IgA molecules that we have produced, we'll have loads of different types that are directed against different epitopes of influenza virus antigens. So overall, we've produced loads of IgG, loads of IgA, and loads of IgM molecules that are directed against epitopes of the antigens of influenza virus particles. So, how are these going to help? We will ignore IgM for now. We'll mainly focus on IgG and IgA because they're the main two forms. And these are both going to end up at the respiratory epithelium. The IgA is going to actually end up on the surface of the respiratory epithelium, and the IgG is going to end up in the exudate all over the place in the respiratory epithelium because it's inflamed at the moment. So they end up in the right place. Now, what do? What is one of the major things they do? Well, of all of these antibodies that we're producing, a lot of them will be directed against hemagglutinin, neuraminidase, and other surface proteins. So we're going to get antibodies that are directed against epitopes of hemagglutinin, neuraminidase, the M2 ion channel, also the MB ion channel in the case of the influenza B virus, and of course in the case of the influenza C virus you don't have hemagglutinin and neuraminidase, you instead have the hemagglutinin esterase fusion protein. So these are the membrane proteins. So we're going to produce a lot of these antibody molecules that are directed against epitopes of the membrane surface proteins. Now what are these going to do? Well, they're going to help destroy virus particles. And how are they going to do that? Well, when you've got a virus particle, and you've got loads of the antibody molecules that can bind to its surface proteins nearby, they are indeed going to bind to its surface proteins. So let me just draw a quick picture of this. And so if I just draw a quick picture of the influenza virus particle, and we'll go for influenza virus A, just because that's the major one. So this is the viral envelope. Let's just put some hemagglutinin proteins in it. So here's hemagglutinin. Then let's put neuraminidase in. So, oops, I meant to change colour. Here is neuraminidase. There's a mushroom here as well. Let's put the M2 ion channel in, like so. And that's it for influenza virus A. So these are the surface proteins. I won't bother drawing everything else in, because that'll just take time and it's not necessary at this point. So, we're going to have antibody molecules that are directed against these surface proteins, and when the virus particles come into contact with these antibody molecules, the antibody molecules are going to bind to them. So let's draw this on. So, I'll draw it on firstly for hemagglutinin here. So, here is an antibody molecule, which it looks like an IgG molecule here, but it could be an IgA molecule. All I'd have to do is put another one on here to make it a dimer. But here is an antibody molecule that has bound to a certain epitope of the hemagglutinin spike there. And the same thing will happen for neuraminidase and M2 ion channels. Now, this is really important. The fact that some of these antibody molecules directed against hemagglutinin are going to bind onto the surface of the hemagglutinin protein, because that's going to make the hemagglutinin protein utterly useless. Remember what the function of hemagglutinin is. It's to bind to the sialic acid residues on the surface of epithelial cells so that it can enter those cells. So if we are now binding IgG and IgA molecules onto the surface of hemagglutinin proteins of the extracellular virus particles, we are neutralizing those virus particles. Those virus particles are being made utterly uninfective, basically. So this is one of the key functions of the antibodies in neutralizing viral particles, so neutralization. And this is specifically or particularly done through them binding to hemagglutinin in the case of influenza A virus and influenza B virus, and through them binding to hemagglutinin esterase fusion protein in the case of influenza C virus. So let me say this again because this is so important. So, we will produce B-cell responses against hemagglutinin and hemagglutinin esterase fusion depending on which type of influenza virus species we are dealing with here. Therefore, we will end up producing IgG molecules and IgA molecules and also IgM molecules that are directed against epitopes of hemagglutinin or hemagglutinin esterase fusion. When the virus is extracellularly, these will be able to bind to the hemagglutinin or hemagglutinin esterase fusion on the surface of the cell, and that will that on the surface of the virus, and that will neutralize the virus. So neutralization of virus. It means that the virus is not going to be able to do anything now. It's not dangerous at all because it can't enter cells. That's what I mean by neutralization. We've destroyed its ability to actually infect cells, and of course that is an incredible success. That's fantastic. And this is one of the major ways in which these antibody molecules are going to attack the influenza virus. And IgG will do that when it's in the exudate at the inflamed site where there will be loads of influenza viruses in the extracellular fluid. IgA will do that on the surface of the epithelium, where of course we know there's loads of influenza viruses continuously budding out of infected cells and then having to go into the extracellular space temporarily. And IgM will do that if any influenza virus particles do happen to go into the blood, they will neutralize them so that they can't do anything. So this is one of the absolutely key ways in which these antibody molecules are going to fight the influenza virus. And indeed, that is the major thing that IgA is going to do. IgM is going to play a minor role because it does not the virus particles do not usually end up in the blood. IgG is going to do this as well. IgG is also going to have some other functions that I'd now like to talk about. So, this one is what IgG and IgA are going to do, and if it does get into the bloodstream, IgM will do that, but that's going to be minor because only a few little virus particles might get into the bloodstream normally. It does not cause a systemic infection, remember. Okay, so what other things then can IgG do that IgA can't do? Well, IgG can also result in opsonization of virus particles. So this is another way in which IgG is going to fight against the influenza virus particles. So opsonization. Now, what does opsonization mean? Opsonization means promote phagocytosis. So when you get IgG molecules produced against any of the surface proteins of your influenza virus, so in the case of influenza A, this is M2 ion channel hemagglutinin neuraminidase. In the case of influenza B, it's hemagglutinin neuraminidase, M2 ion channel, and NB. In the case of influenza C, it's hemagglutinin esterase fusion and CM2. When you get any IgG molecules produced against surface antigens of the influenza virus particle, they will bind onto the surface of those. And we've talked about neutralization of the hemagglutinin esterase fusion protein, but now also when you've got a virus particle floating around in the extracellular fluid that is covered in IgG molecules, that is a giveaway sign to phagocytes such as neutrophils, macrophages, and dendritic cells that this is a virus, that this is a pathogen. If you ever have a structure floating around in the extracellular fluid that is covered in antibody molecules, that is a giveaway sign that that is a pathogen. Now, previously it was very difficult for them to recognize influenza particles. It was difficult for them to phagocytose the influenza particles because it was difficult for them to recognize that the particle was absolutely, most definitely a pathogen and needed to be destroyed. Now, if they're covered in antibody molecules, it is an absolute giveaway sign that the influenza particle is a pathogen and needs to be destroyed. So let me just put this in here. So we'll have our macrophage as the classic example of a phagocyte. So here is our macrophage, our M5, and then we'll have our influenza viral particle here in red. So if this is now covered in antibody molecules, and I'll just do these in blue, so these little lines are representing antibody molecules. If it's covered in antibody molecules of the IgG class, macrophages have on their surface a receptor for IgG, and so do other types of phagocytes such as dendritic cells and neutrophils, and that receptor is called the Fc gamma R3. So Fc gamma R3. Now what does this mean? This refers to the constant region, the Fc region of the antibody molecule. Gamma refers to IgG. Remember, gamma is Greek and it's kind of equivalent to G. So the constant region of IgG, it's a receptor for the constant region of IgG, and it's the receptor number 3. So on the surface of phagocytes such as macrophages, neutrophils, dendritic cells, you have this Fc gamma R3 receptor, which is a receptor for the constant region of an IgG molecule. So when you have anything floating around in the extracellular fluid that is covered in IgG molecules, they will bind onto this receptor on the surface of the phagocyte, and then the phagocyte will know that this is a pathogen because it's coated in antibody molecules, and nothing that is healthy and normal should ever be coated in antibody molecules. And therefore, phagocytosis of free virus particles is made much easier once you've got the antibody molecules marking the pathogen particles out. And that process of promoting phagocytosis of pathogens is called opsonization, and that is a function of IgG. Now, it's only IgG because this receptor, the Fc gamma R3, is a receptor for the constant region of IgG, not IgA, which is why the IgA isn't going to do this. Of course, the virus particle might get covered in IgA as well as the IgG, but as long as it's got lots of IgG molecules on there, those will be the ones that promote the opsonization. The IgA is just there on the side, basically. It's not going to promote the opsonization in the way IgG will. So that's another way that antibody molecules can promote the destruction of the virus, the wiping out of the virus from the human body. Okay, and then one final way that antibodies are going to really help with the destruction of the influenza virus is going to be through a mechanism called antibody-dependent cell-mediated cytotoxicity, which for short, is abbreviated down to ADCC. So this is the antibody, which I'll short to AB, and then dependent is the D, so antibody dependent, and then the first C is for cell-mediated cytotoxicity. So this is a mechanism of killing virally infected cells, another mechanism of killing virally infected cells that is antibody dependent. So we have seen quite a few mechanisms of killing virally infected cells. We've seen the natural killer cells can go around looking for low MHC class 1 molecules on the surface of cells, and if they find significant enough lack of MHC class 1, they'll know that there's something dodgy about the cell and they will kill it. We've seen the cytotoxic T lymphocytes, which have this incredible way of seeing whether cells are virally infected by looking for the peptide fragment that they are directed against on the surface of the cell bound to MHC class 1. And we're about to see another mechanism for killing virally infected cells that is natural killer cell driven. So, the cytotoxicity, the death of the virally infected cells, is mediated by other cells, and those cells are actually going to be the natural killer cells again. So this is another function of natural killer cells, so the NK cells are going to do this, and it's going to be antibody dependent. So remember, natural killer cells need very basic signs that a virally infected cell is indeed virally infected. One of them is MHC class 1 going down. We discussed that long ago. Another one would be if a cell becomes coated in antibody molecules. So we've just talked about how if a free pathogen becomes coated in antibody molecules, that is a giveaway sign to neutrophils, macrophages, and dendritic cells that that thing is a pathogen and therefore that it should be phagocytosed and destroyed. What if a virally infected cell becomes coated in antibody molecules? Now, why would they become coated in antibody molecules? Well, there is a reason. Think back to the viral replication cycle. The virus puts all of its proteins on the surface of the virally infected cell. So if this is a virally infected cell here, so again, I'll put the red cross for it. Remember, on its apical surface, it's going to have loads of the viral proteins in it. It's going to have hemagglutinin, neuraminidase, the M2 ion channel, all of the membrane proteins of the virus are put into the cell membrane, the apical cell membrane of the infected cell, into those clusters so that the virus can bud off the surface of the infected cell and will end up with a nice viral envelope with the viral surface proteins in. So, infected cells do end up with the viral antigens on their surface, and therefore what can happen is these can end up antibody coated as well, coated by IgG and IgA, of course, the two antibodies that are going to be at the site of inflammation. So I'll just put this on. So here are these loads of little blue spikes again all over the apical surface here that we will imagine are bound to hemagglutinin, neuraminidase, M2 ion channel, all of the surface proteins of the influenza virus. Okay, so they're bound to epitopes of surface influenza viral proteins. So this is again a giveaway sign that this is a virally infected cell. Now the phagocytes, they would struggle to phagocytose an entire still alive epithelial cell. They do not like phagocytosing entire human cells. When the cell has died, what they will do is they'll phagocytose little bits of it, because remember, when the cell dies, it will break up into loads of little pieces, and it's the little pieces that those phagocytes are phagocytosing. So when I've previously been talking about how macrophages, dendritic cells, and neutrophils will phagocytose dying epithelial cells, they do not phagocytose them in one great gulp. These cells are too big. The cell has broken up into loads of little chunks, and they are phagocytosing the little chunks of the cell. Okay? So phagocytes cannot destroy this entire great big virally infected cell. You need something else that is designed for killing human cells that are dodgy, and natural killer cells are just the man. Okay, so let me put in our natural killer cell here. So here is our natural killer cell, the round cell with the large nucleus again. Now, remember, natural killer cells, they had the KIA receptor, the killer cell immunoglobulin-like receptor for looking for too low MHC class 1 molecules. They are also equipped with this receptor that we've just been discussing, the Fc gamma FR3 receptor, and therefore they can bind to the FC region, the constant region of immunoglobulin G molecules, IgG molecules, on the surface of these virally infected cells. And when they see that, they know that's a signal that this cell is virally infected, and they unleash their perforin and granzyme mechanism on this cell to try and trigger the apoptosis mechanism in it, and therefore destroy the virally infected cell. Okay, so we have completed our discussion of the adaptive immune response. We will have a break here because we've been going for 1 hour and 10 minutes now. In the next video, I will summarize the adaptive immune response because I think it is worth repeating it one final time to make sure that this is nailed in, because it really is important to understand this. Then we'll talk about how you get long-lasting immunity to influenza virus strains, so how you get a secondary immune response and things like that. And then we'll talk about antigenic drift and antigenic shift of influenza virus strains.